Hello students, welcome to GDC classes. So in this today's video, we basically talk about the rest of the part of the sterilization topic. So as we discussed, what is sterilization? So sterilization is a process by which we completely remove a viable microorganism from a particular area. So last time we are talk about different type of sterilization. Let's summarize what we have studied from the last video. So if we talk about there are different methods, there are two major methods for the sterilization. One is physical method and another one is chemical method. So in physical method, we heat, filtration, cold treatment, such type of condition is what we have discussed. So let's summarize. So if we talk about heat, heats like moist heat. So moist heat, there are divided into two type, boiling or running, which is basal mechanism working is denaturation of the protein present in the microorganism. This is the most effective way to kill the bacteria or fungal pathogens. So equipment like basins, pipelines, SS joint, SS walls, SS forms can be sterilized. Next one is autoclaving. This is one of the most important process where both temperature and pressure working simultaneously on the particular on microorganism on the major mechanism of action is denaturation. So extremely effective method at 15 psi pressure around 121 degree degree temperature kills the almost vegetative cells and their corresponding endospore in just 15 minutes. Okay, so this methodology for the used for the microbial culture media sterilization solution dressings, utensil, dressings, lines, linens, which can be which can stand the pressure and temperature which is generated in the autoclave. Another method one is pasteurization. So pasteurization is basically carried out for the pathogens pathogens present in milk. Okay, so it's heat treatment of fresh milk at 72 degree centigrade per 15 seconds, which kills the pathogen plus other non-pathogen as well. So milk, cream, along with some fermented alcoholic beverage can also be uh, sterilized by this method. If we talk about dry heat is also subdivided into three classes. One is direct flaming, incineration, hot air sterilization. So direct flaming contaminate are burned to ashes, most effective method for sterilization. Okay. Next is incineration, burning to ashes. Like whenever we have seen that some of the medical wastes, like things when they can contaminate to other things like gauze, cotton, and some of the stitches, bandages. So that's what we have to do is we have to burn into ashes. Very effective. This is the very effective means of sterilization. Contaminated dressings, wipes, bags, and paper cubes. Next is hot air sterilization. So hot air sterilization majorly act on the mechanism of oxidation. This is very important from exam point of view. So question can be asked like autoclave is worked on the principle of denaturation. Similarly, hot air sterilization is worked on the principle of oxidation. Extremely effective means of sterilization requires heating at 170 degree centigrade for 2 hours. 170 degree centigrade, you have to heat for at least 2 hours like your surgical instruments, dental instrument, needles, empty glasswares, and glass syringes can be sterilized. Next, we will talk about filtration. So filtration is a process by which, by using up a typical filter, which is called as membrane filter. We called it as a membrane filter having diameter 0.22 micrometer diameter bore size. So we have to place and we have to separate the pathogenic microorganism from particular solution. Like the filtration process is basically applicable for the solutions like antibiotic solutions, antibiotics, vitamins, steroids. Okay, such type of preparation. Liquid or gases made pass like a screen-like material which traps the bacteria. Common filter used in consist of the filter which is basically consisting of nitrocellulose or cellulose acetate. Okay, invariably used for the liquid samples such as vaccines, toxins, and enzymes. Next is cold treatment. Whenever we know that whenever we reduce the temperature to 0 to 7 degrees centigrade, so there are 0 to 7 degrees centigrade. So what happens? So there will be a reduction of the reproduction of certain pathogenic microorganisms. So there are three types of cold treatment we can give. One is refrigerator, normal refrigerator, which is there in our home. One is deep freezer, where temperature can be reached to maximum up to 95 degrees centigrade, and another one is lyophilization. So basically, they reduce the chemical reaction and probable changes in the protein. This is basically a bacteriostatic process. Okay. Another one is deep freezing. This is very effective for preserving microbial culture, like microbial culture, or like in during biomedical researches, whenever we are working on any animals or humans, so we have to keep their sample for prolonged period of time, like organ sample, like liver, kidney, we have to preserve it for prolonged period of time. In such cases, what we have to do is we have to keep the sample in the deep freezer. That is around one freezer is coming, which is called as -80 freezer, which we call it as a deep freezer. We kept the sample over here. Okay, another one is lyophilization. Lyophilization is a long-term preservation. If you want to preserve the sample for a year or for months. So what we have to do is one process is there that is called a lyophilization, which can be store the sample up to months to year. Next is desiccation, where absolute destruction of the metabolism in the absence of water. Okay. Next, next is osmotic pressure. As I told you, osmotic pressure, like whenever we are preparing any pickles in our home, we add more amount of salt or sugar into that pickles to for prolonged period. This is basically act on the principle of osmotic pressure. This basically causes affordable loss of water from the microbial cells. Okay. Next is radiation. There are two major type of radiation is used. One is ionizing radiation. Another one is non-ionizing radiation. If we talk about ionizing radiation, so ionizing radiation basically destructs the DNA. So they basically destruct the DNA by gamma rays, high energy electron beams. This is not a common in routine sterilization, but extensively used for the sterilization of pharmaceutical products, plus medical and dental supplies. Okay, like basically your pharmaceutical products, dental products sterilization. Next is non-ionizing sterilization. This is causes permanent DNA damage. Okay, non-ionizing radiation basically causes permanent DNA damage to the microorganism by UV light or UV radiation. This is overall radiation is not very penetrating. So common microbes in a closed environment using UV lamps are basically used. Next is about what are the biological indicators that are used for the different type of sterilization. What are the biological indicators that is used for the different type of sterilization? If we'll talk about the indicators, so for dry heat sterilization, the biological indicator is Bacillus subtilis, and the chemical indicator is Brown's tube and Bois dictates. This question was asked in GPAT 2015. Next is moist heat. So moist heat major mechanism work is denaturation of protein. Okay, denaturation of protein. Okay, so the biological indicator is Bacillus streptothermophilus. Biological indicator is Bacillus streptothermophilus. In this was a question was asked in GPAT 2012. Other indicators are Bacillus subtilis, Bacillus coagulans, and Clostridium sporogens. Okay. Next, you will talk about gaseous sterilization. The biological indicator is Bacillus subtilis, and the chemical indicator is 𝘂𝙎. Chemical indicator is 𝘂𝙎. Next is you will talk about radiation. For the radiation, biological indicator is Bacillus pumilus and Bacillus capricius, and the chemical is chemical densitometer. If we talk about filtration, the biological indicator is Pseudomonas diminuta, and physical indicator is bubble point and pressure test. Okay, these are the typical biological and chemical indicator or physical indicator which is used for the different type of sterilization method. Like that means that there are this particular biological species for this particular bacteria is used for the indication of dry sterilization or moist sterilization process. We will talk about chemical use for sterilization process. We have discussed all the physical method of sterilization. Now we basically talk about chemical method. So in the chemical, first is phenol. and phenolic derivative. So if we talk phenol, or we also called it as a carbolic acid. Okay, plasma membrane. So it basically acts on the mechanism of plasma membrane disruption, enzyme denaturation, and inactivation. Not so frequently used, but rarely used it as a antiseptics or disinfectant because of its disagreeable odor and irritating features. If we talk about phenolics, these are for the instrument. This is for the instrument, mucous membrane, skin surface, and environmental surface. So for these purposes, these phenolics are used. Orthophenyl phenol and hexachlorophene and phenol are derivatives. These are basically used in case of different prepare marketed formulation like Dettol, Savlon, in some formulation. These are used as a component. Next is chlorohexidine. So chlorohexidine basically acts by disrupting the plasma membrane. So degerming the skin, specifically surgical scalps. Okay, basically for the in case of surgical process, this is basically used. So extremely bactericidal. This is basically bactericidal action on gram positive as well as gram negative microbes and non-toxic persistence. If we'll talk about halogen like iodine, chlorine, iodine inhibits the protein function strong and it is a strongly oxidizing entity. Similarly, chlorine form a strong oxidizing agent, hypochlorous and hypochlorous acid that changes the cellular constituent. So iodine tincture, as you know, the betadine solution iodine tincture replaced with iodophorous chlorine gases to district water supplies. Okay, iodine I2 and chlorine C2 are act either individually or in a combination as a potent organic and inorganic compound. Next, if we talk about alcohols, so alcohols basically lipid or fat dissolution protein and denaturation. So this is clinical thermometer swabbing such type of things like 70% alcohol is used, 70% ethanol basically used for the cleaning like your surgicals, your thermometers, your tables, where the basically surgical process are carried out. This thing for cleaning of this thing, this is basically used. This is a potent bactericidal and fungicidal, not effective against non-involved virus and endospores. Okay. Next chemical is heavy metals and their derivatives. So they basically denaturation of enzymes and other essential protein of microbes. So they what they do, they basically denaturate the enzyme and the other essential proteins present in a microbe. Similarly, silver nitrate prevent the genococcal eye infection, meibomian dysfunction, skin mucous membrane. Okay, silver, mercury, copper usually serves as a germicidal, antiseptic, algacidals. Next, surface active agent like soap, acid, ionic detergent, acid, anionic detergent, and cationic detergent. So you all know that we are different in different work, but we are basically apply soaps in our hands for the cleaning of microbes. Okay, for that, what they do, they bacteria remove the mechanically by. So when we rub the soap, like if this is a soap, we basically what we do, we basically rub around our hands. So during the rubbing process, what happens? These microbes are washed out from our hand. Okay, that leads to enzyme disruption or inactivation takes place. Protein denaturation like enzyme inhibition, disruption of plasma membrane can be seen. So like trichloroban, teprol, Zeprian, Sepacol are also used for these purposes. Next, we will talk about organic acid this metabolically inhibition of largely affecting molds. So, like what are the used? Benzoic acid or sorbic acid basically. Next, we will talk about aldehyde affordable protein inactivation. So, they basically inactivate the protein present in the microorganisms. So, Cydex is one of the brand name which contains glutaraldehyde is used. Next is gross chemosterilizers. So they basically acts on the principle of denaturation. They superb and excellent sterilizing agent. Ethylene oxide is employed. So ethylene oxide is used in that gaseous sterilization process. So you have to remember what chemical is used for what, which one is used for gaseous, which one is used for chemical, which one is used in soap, which one is a organic acid or alcohol or aldehyde. You have to remember. Next is oxidizing agents. Ozones and hydrogen peroxide are used for as a oxidizing agent. So that's all for that is all about we discussed about the what are the different type of sterilization methods, what are the application where they basically employed. So in today's video we basically talked about what are the chemicals used for the different sterilization process, their mechanism of action, how they can be employed in particular sterilization process. We have discussed in detail. So in the that's all about your sterilization topic. Thank you.